Good afternoon and welcome to Atlanta Rotary. Happy Valentine's Day to all of you. Bob Hope had threatened to dress up as a Valentine today, so we had to spice things up a little bit to head that off. Hope you all had a wonderful weekend, and congratulations to the LA Rams for their Super Bowl win last night. What a fun game to watch. Such a wonderful game. And also a big congratulations to Alana Myers-Taylor. You may remember us showing a video of Alana Myers-Taylor a week or two ago. Her daughter, Rotarian Eddie Myers, is a superstar in his own right. And Alana last night won the silver medal in the women's monobob. We are so proud to consider her part of our extended Rotary family and look forward to cheering her on as she heads on to the women's two-person bobsled later this week. We have another great program planned for you today, and I will let us kick that off with Doug Hooker, who will join us to lead us in the Invocation and Pledge of Allegiance. You will actually see Doug again in a few weeks, March 21st, when Doug and author Andrew Filer will cap off our celebration of Black History Month. They will be telling the story of the Rosenwald schools, 5,000 schools that were founded for African-American children in the South in the early 1900s. Doug will be sharing music from his symphony that he just wrote. The symphony just had its world premiere last weekend. And Doug, you are quite the Renaissance man. And so we're so excited to have you with us today as well. Welcome.
Thank you. Thank you, President Catherine, and happy to be here. So if you want me to take it away, I'll do that.
Yes, and I'll turn it over to you.
Okay. We will start with the invocation, and then I'll lead us with the Pledge of Allegiance. So, ladies and gentlemen of Rotary and your guests, if you will now join me in an attitude of prayer, if you're able and willing, I will begin. Gracious God, our Father, God, our Mother, our sister, and our brother, you who are the universal spirit that speaks to the good in all of us. This Valentine's Day, help us to realize that love is deeper than the romantic love we often equate with this day. Love is the universal force which enables us to connect across the seeming divides of race, religion, creed, gender, sexual orientation, and more. Help us to realize that true love can enable us to connect human to human and learn how to respect one another and learn how to live peaceably with each other, no matter what we personally believe. As we gather now for this meeting and this meal, remind us to be grateful for the blessings represented by the food we partake and the fellowship that Rotary provides. Remind us to be grateful for the many people in the world who help to improve our lives even when we are not aware of it, like our speaker today. To be grateful for Dr. Dennis Liotta and his colleagues who are helping us to fight this pandemic in their own way with the skills and the experiences that you have blessed them with. And so now, as we partake of the food and the fellowship and the new knowledge that we will experience today, may we be reminded that all of it is a blessing, and may we use all these blessings in your service. Amen. And now, ladies and gentlemen, the Pledge of Allegiance, if you will join me. I pledge allegiance to the flag of the United States of America and to the republic for which it stands, one nation, under God, indivisible, with liberty and justice for all. Thank you all very much.
Thank you so much, Doug. Such beautiful words. And we look forward to seeing you again in just a few weeks. Thank you.
Thank you.
Wonderful. A reminder to please sign in using the QR code that you will see pop up here on the screen in just a minute. And welcome again to all of you joining us today. I think we have a slide with that QR code. And if you don't have the QR code or would prefer, there's also a link below the video screen that you can use to register your attendance as well. Quick agenda preview following our announcements. We will have new member Simon Malco introduced by John Yates. And then we continue our public health series with a fascinating program headed up by Emory professor Dennis Liotta, whose lab recently released the world's first pill for COVID. Looking further ahead, we'll be off next Monday for President's Day, and then we return on February 28th for the exciting finale of our series on public health, when we will host Dr. Rochelle Walensky, director of the CDC, who will have a very special visit with our own Atlanta Rotary Club. Upcoming programs also include Mayor Andre Dickens, Atlanta Fed CEO Raphael Bostic, a 4-part series on the supply chain with 4 notable CEOs in that space, and even a visit to the Norfolk Southern headquarters for one of our Rotary on the Road events, where we get to visit the nerve center for the entire railroad operations for Norfolk Southern. Just a fantastic series of programs. I know you won't want to miss a single one of them. Finally, also a reminder that we will return to in-person programming on Monday, March the 7th. We cannot wait to see all of you then. Thanks also to all of you who have registered for our small group dinner series. We have had a tremendous turnout. As you may have heard previously, these are informal dinners with about 8 or 10 Rotarians gathered, very casual, but just joining together for conversation and fellowship during the months of March and April. If you have not already signed up, please do so. The deadline is tomorrow, February 15th, and you can find the registration link in your program reminder email. Invitations go out tomorrow also for our annual Old Timers Dinner. This is going to be a fabulous evening. So excited about this, March the 9th at the Capital City Club. For any of you who may be new to the club, the definition of old timers is really any member who has been a member of the club for 20 years or more. That does not necessarily mean, by the way, that you are old. So please join us for that if you qualify as an Old Timers member, and really special thanks to Tommy Hills, who's done such a great job organizing this event. Congratulations also go out to Kathy Waller, our very own Rotary Club treasurer. Kathy was recently named executive director for the Atlanta Committee for Progress. She follows in the footsteps of another Rotarian, Shan Cooper, as she assumes this new role. Kathy was previously CFO and controller of the Coca-Cola Company, and we're so excited to collaborate with Kathy in this new role. ACP has always been a great friend to Rotary, and we wish Kathy all the very best in this exciting new role. Finally, last but not least, please join me in congratulating all of our Rotarians with February birthdays. You'll see their names there on the screen. So excited, and I wish all of them a very, very happy birthday from Rotary. I'm now pleased to introduce fellow Rotarian John Yates, who will introduce new member Simon Malco. John, it is always great to see you. I know you are already warming up in the sort of president on deck following President-elect Stephanie. They always give us plenty of time to get ready for these things. But you do such a great job bringing us new members and introducing great programs. So you've done it again today, and we really appreciate your introducing us to Simon. So welcome.
Well, thank you, President Catherine, and happy Valentine's Day to you and all the members. Along with my co-sponsors, Katie Kirkpatrick, Richard Kopelman, and David Rubinger, I'm pleased to introduce the club to Simon Malco. Since 2018, Simon's been the managing partner of Morse, Manning & Martin, where he's the youngest managing partner in the firm's history. Earlier in his career, Simon served as the firm's litigation practice chair and also was general counsel to the law firm. Under Simon's leadership, Morse, Manning & Martin has become one of the fastest growing law firms in Atlanta and has developed a national reputation in areas such as real estate, technology, and corporate finance. Simon has also spearheaded the law firm's strategic plan, which has seen the firm's revenue increase by almost 25% during his brief tenure as managing partner. It's also thanks to Simon's commitment to the community that the firm's outreach committee has started programs in areas of civic leadership, diversity, pro bono service, and women's initiatives. And I'm particularly thankful for Simon's support of Cure Childhood Cancer. Like so many of us, Simon is a transplant to Atlanta. His family moved to town when he was 13. He graduated from Dunwoody High School, then Emory University, and Fordham University Law School. And right after law school, Simon practiced law for 8 years in New York. He saw the light in 2002 and moved back to Atlanta to join Morris, Manny & Martin. Simon's also a member of the board of the Metro Atlanta Chamber and remains an active civic leader in our community. He's married to Christy Malco, and they have 2 wonderful children, Grace, age 16, and John, 11. Simon's hobbies include coaching, playing, and watching soccer. As you see on the screen, he has a very interesting avocation. He's a certified volunteer service dog handler and trainer. And Simon, I'm sure these are also skills that come in handy in managing a group of hard-charging attorneys. So I'm proud to call Simon my friend and colleague. Please join me in welcoming Simon to the Atlanta Rotary Club and congratulate him in person at our March meeting. Thanks. And I think you're muted, Simon.
Thank you, John. Thank you for that kind introduction. You're a wonderful colleague and a good friend. I'd like to thank my sponsors, David, Richard, and Katie. And I look forward to meeting all of you in person at our next meeting. So thank you very much.
Simon, welcome. Welcome to the world's greatest Rotary Club. I love the background that you have as a service dog handler. I know that is such an important role and really important thing these days. And very impressed that you have those skills. Also, as a mother who has spent many hours on the soccer field with my own kids, I truly appreciate your mentorship and your leadership of our young people. So again, welcome to Rotary. You're going to love being part of this organization, and we truly appreciate your leadership in this community. Look forward to seeing you soon.
Thank you very much.
Terrific. So as you all know, over the past few weeks, we have spoken several times about the seismic changes occurring in the public health sector and the critical role that Atlanta has played as a global leader in that sector. Today, we're extremely fortunate to have with us the person who has been leading the lab that has been a world leader in developing antiviral treatments. Our speaker today, Dennis Liotta, is going to give us a behind-the-scenes look at the world of drug development. David Rubinger, co-chair of our program committee, will lead us in that conversation, and we'll be introducing Dennis to the group today. David, as we all know, is market president and publisher of the Atlanta Business Chronicle. Welcome, David.
Welcome, President Catherine. Thank you.
Thank you so much for your continued leadership and for, as you always do, asking fantastic questions and really helping us understand and dig into this fascinating topic today. So I'm going to turn it over to you to take us forward.
Thank you again, President Catherine. I really look forward to today. Before I turn it over to Professor Liotta, I just want to say one thing. We mentioned the LA Rams winning the Super Bowl. I wanted to make special mention of one of their players was Grant Haley, who many Rotarians may have known. Grant Haley's father, Dr. Leon Haley, who headed up emergency medicine at Grady and had been at the University of Florida Medical Center, who passed away last August. It warms my heart as another connection to Atlanta that Grant Haley was able to win the Super Bowl. It must be bittersweet for him and his family within the year of his dad's passing. But I thought about him, and I thought that was a special moment for those of you who knew Dr. Leon Haley, know that his son is one of the Super Bowl victors, which I thought was really nice. But again, as President Catherine said, this is our month of talking about critical healthcare issues, and I'm so excited that Professor Dennis Liotta is with us today. And I want to give special thank you to Russ Lapari of Health Connect South. He was one who connected me to Dr. Liotta to have this program. And Dr. Liotta is one of those special people in our community that not enough people know about, in my opinion. He founded and is the executive director of the Emory Institute for Drug Development. Together with the Drug Innovation Ventures at Emory, which you're going to hear more about, Drive, which he also founded, EIDD discovered the drug that is now on track to become the first oral drug for combating COVID-19. But But this is just the latest in Dr. Liotta's amazing career. He's got a long distinguished career at Emory, and he's made significant contributions to improving human health and touched the lives of millions. His long-lasting legacy in technology and innovation is manifested on so many different frontiers. Over 100 issued patents in the US alone, more than 20 patent families currently under prosecution. At least 18 life-saving FDA-approved therapies originated from these innovations. And this is my favorite one, more than 90% of people in the US with HIV/AIDS take at least one of the drugs invented by Dr. Liotta. So his impact and his team's impact on the drugs that save millions of lives reaches far and wide, and his journey is a great story. So with that, Dr. Liotta, I'm going to turn it over to you. Thank you so much for being part of our Rotary today, and look forward to the conversation.
Thank you, David. And thank you all for this chance to chat with you. I hope I don't go on very long, so we'll have the chance to have a dialogue afterwards. My title, you can see, From HIV to COVID-19, and I want to tell you about my lab. I'm going to try and keep the strategic aspects of our work at reasonably high visibility, and I'll slip in a little science along the way just because I couldn't resist. Could I have the next slide, please? So since I'm an academic researcher, I always worry about people's perception of what actually goes on in academia. So I decided to look up the two terms and share the definitions with you. So if you look at research, it's a pretty noble task. It's a careful or diligent search, studious inquiry or examination. It's the kind of thing that most people would be proud to be associated with. However, when you look up academic, you get a kind of mixed message, associated with the academy. But when you get a little further down, the first red flag is very learned but inexperienced in practical matters. That's not terribly exciting, but at least they did put it parenthetically thinkers. And I said, okay, that's good. When you get further down, it gets a little more questionable. Item 4B, having no practical or useful significance, with the synonym pedantic. And a long time ago, I decided that if I'm going to work really hard on my research, I want to be sure it is both practical and significant. So I'm going to give you a little overview of some of the things that we've done. Can I have the next slide, please? So as David mentioned, we've been very fortunate that we've gotten a lot of FDA-approved therapies associated with at least some of the drugs that we've prepared. And the 17 approved combination therapies for HIV, and you know that in HIV, we only use combination therapies, never monotherapies. They've all at various times played an important role in advancing our ability to treat this disease. But I wanted to focus just for a moment on this one triple combination called a tripla, and I'll illustrate it on the next slide. So if you go back to the mid-90s, when combination therapy started to become the way to treat HIV infection, it worked, but it was extremely cumbersome. So if you look at the hand on the left, many patients were required to take 15 to 20 pills a day. And some of these were before you ate, some of these were after you ate, some of these were in the middle of the night. Many of the early protease inhibitors that you might have heard about had to be taken every 8 hours. So if that meant you had to take it in the middle of the night, then you had to wake up. And if you didn't, then the levels of drug in your body could go below a critical point where they were still protecting you. And once they dropped below that level, then viral replication could occur, which meant the chance for mutations that would make your drug either less effective or completely ineffective. So going to a tripla where we had 3 drugs embedded in one pill that was taken once a day was truly a game changer. And as David mentioned, this has been very impactful, and over 90% of HIV patients on therapy take or have taken one of the drugs invented in our lab. So this was really a very nice, satisfying story. The less satisfying part of it, however, was that it took 13 years to move this drug from conception to actually being in the marketplace. And that that is just too slow for those of us who are trying to make a difference in the health of the public. My colleagues and I decided we needed a faster, more efficient way to move things along in the academic setting. I can't control what goes on in the commercial setting. Next slide, please. So just a couple of key points. Drug development is an expensive and time-consuming process. It can take anywhere from 5 to 15 years to develop a drug, and it can cost hundreds of millions to billions of dollars to do this. So this is an undertaking that's not for the faint of heart. And what this slide illustrates is one of the issues that we have. If I wake up tomorrow morning at 3 o'clock and have this really great idea, and I call up one of my friendly venture capitalist friends and say, look, I just had this fantastic idea, and I wonder if you could give us some money to help bring this along. He or she would undoubtedly say, listen, it sounds interesting, but as far as we're concerned, that is a total risk idea, 100% risk. And give us a call when you've figured out how to de-risk this sufficiently so that we'll put some money into the development process. Well, in academia, that is sometimes problematic. What we try to do is lower risk and add value, but do it at reasonable cost. So we can't get into the hundreds of millions or billions of dollars, but we can, through government grants or other sources of funds, we can make some progress. The valuation of our technology as we go forward is not linear, which can be sometimes a little problematic to have to deal with because it's actually a step function. And so we can go along and do a lot of work, spend a fair amount of money, and only after a certain point, a certain milestone, can we claim to have a better valuation. So that's part of the reality of doing drug development in academia. Next slide, please. The remaining reality is something that we affectionately refer to as the valley of death. And this is the point at which you're far enough along in the development process that government funding gets very difficult to acquire. We have, for discovery, there's a lot of opportunities to get funding, But for development, it's less sexy. It's pretty much heavily prescribed in terms of what you have to do. And funding agencies don't usually get very excited about doing necessary, but what they consider routine advancements. So the sources of money fall off. And usually at that point, it's too early for the private sector to feel that the risk has been sufficiently lowered to put their funds in. On top of that, there's a number of problems. Scientists like myself are trained to do science, and we're trained to do discovery, but development is another kind of operation entirely. And typically in the academic sector, we lack experienced leaders and managers and people who just understand the development process. Secondly, another stereotype you might think about in terms of academics is we're not exactly known for our ability to make nimble decisions. In the worst case scenario, when a problem arises, the typical approach in academia is to form a committee and let them study it. Well, obviously, a private sector company, especially a small startup, something like that isn't going to work. You have to be able to make decisions on the spot and move things forward. Finally, something that most people don't know, every research university has an intellectual property policy, and embedded in that are revenue sharing terms. And it turns out that most of the revenue goes that's obtained, let's say when you achieve a milestone in a license, goes to inventors departments, inventors schools, but not into the project itself. So if you had a biotech company and you were moving something forward and you got some money, you would plow it back into the enterprise. In academia, unfortunately, a lot of these things get distributed around, and it would be a very hard process to try and overcome. So in order to do this and to find a way to expedite it, we created something we call the DRIVE model. And I show you that on the next slide. So DRIVE is an acronym that stands for Drug Innovation Ventures at Emory. And this model has two components. DRIVE, which is kind of the business development end of the partnership, and then the Emory Institute for Drug Development, which are the scientific partner of DRIVE. So DRIVE is a wholly owned subsidiary of Emory, but it's a separate legal entity. And as such, all of those rules that I was just describing don't apply. So this was our way of just circumventing all of those embedded and unintended impediments that exist in academia. And we brought in a small cadre of people from the commercial sector, people who had extensive biotech and pharma experience and who had actually led the discovery of multiple antiviral drugs. And then we use their expertise and a number of strategic alliances that we had made along the way to actually bring drugs forward through the development process when we didn't have either in-house capabilities or strategic partners. Then we would simply outsource to contract research organizations to do whatever else we had to do. And then the EIDD, where this is a cadre of scientists who did the actual scientific investigations. And this partnership has worked very well. So the idea is we take in therapeutic opportunities either from Emory sometimes from my lab or from other sources. We'd use our funds or we'd acquire outside funds to bring these along the development process. And then when we've reached a suitable value inflection point, we would either pursue a third-party license for them to complete the development, or in certain cases, create a biotech spinout to bring the technology forward. So it turned out to be a fairly good model. Another nice subtlety associated with it is, although we're a separate legal entity, we're part of Emory, which means, unlike a typical startup in other arenas, we have ready access to all the physical and intellectual resources at Emory. This gives us a great advantage to use these, rather than if you're completely separate to try and have to pay for those kinds of services. It's really a much better way of doing this. And I thank the administration at Emory for supporting this model. And we got it going right in 2012. And we seeded it with with about $10 million in some space and equipment from the university, and took it forward. Next slide, please. One of the things that we decided to do, $10 million in one sense sounds like a lot, but in the context of hundreds of millions of dollars that it takes to develop a drug, it's not very much. So it was a starting point that we used to advance these compounds, but also to find ways of getting additional money. Because the amount was limited, we decided to focus on what we thought was the highest probability event. And this turned out to be quite fortuitous. So all our expertise was around the development of antivirals. And when we did the research, it was clear that the predominant cause of epidemics and pandemics were RNA viruses. And in fact, these RNA viruses actually account for about 80% of the viral disease burden worldwide. Layer on top of that when we started, there were actually no drugs, zero, available to treat any of these RNA viral infections. So it seemed to us that this is something that had to eventually happen. Eventually, an epidemic or pandemic associated with a single-stranded RNA virus would emerge, and hopefully we would be well positioned to do this. One of the things that differentiates us from, say, a biotech or a pharma company is that they will only pursue drugs where there's a guaranteed market, because that's the only way that they can get investment dollars into their operation. We, on the other hand, were able to use various sources of money to pursue these viral diseases. And in that way, it placed us ahead of the curve. Important to the strategy, however, was that we wanted to try to find drugs that were broad spectrum drugs. And this is not so easy to do with viruses. With antibiotics, we do this all the time. We have broad spectrum antibiotics, and they're very common. With drugs, in viruses, it's mostly what we call one bug, one drug situations, where you develop it for a specific virus, and that would have been opposed to the strategy that we set. So we were going after RNA viruses because of the large viral disease burden, and we were going to try and get broad spectrum drugs so that we have a better shot at being in the right place at the right time when a pandemic does occur. And this was in 2012 and 2013 when we came up with this strategy. Next slide, please. One of the compounds that emerged was coded, as we show here, EIDD 1931, and it did show activity against a variety of virus families, and you can see on the right some of the diseases that it affected. Up until January of 2020, of course, we had no idea that it was effective against SARS-CoV-2 because we were just beginning to hear about SARS-CoV-2 in January of 2020. And in fact, we had brought this compound along as a potential treatment for influenza. We had brought it to the point where we had filed an IND, an investigational new drug application, with the FDA, and we were in position then to take it forward and begin clinical trials. And those clinical trials were being paid for by an NIH contract. And when the realization of SARS-CoV-2 hit everyone, NIH allowed DRIVE and EIDD to switch this from influenza to SARS-CoV-2 because we had demonstrated in cell culture and in some early stage animal studies that this compound showed activity against it. So we were off and running. Next slide, please. I mentioned in the beginning of the presentation that HIV only is we only use drug combinations to treat HIV, and that's because single drugs oftentimes develop resistance fairly quickly, and that was going to be a problem for any pandemic. But one of the things that we particularly liked about EIDD 1931, and we were pleasantly surprised as we studied it, to find that actually it had a different mechanism of action. It confused the viral polymerase as it was trying to replicate the viral RNA. And without delving into the chemistry here, the polymerase, because we could shift around a hydrogen, sometimes this compound looked like one of the building blocks of RNA, uridine, and other times it looked like another building block, cytidine. And so when these are being incorporated into the growing strand of RNA, a lot of mistakes occurred. So if you think of this as some engineer who is has a train that he or she is trying to put together, and it's got a yellow car and a blue car and a green car and a red car, and the engineer says, okay, I need a red car, now I need a yellow car now. And this is the same thing with the RNA bases. I need a uridine, I need a cytidine. But if they get confused, that leads to mismatches. And those mismatches ultimately lead to what we call viral ablation or just cell death. So infected cells, due to these catastrophic mistakes, they simply die. That's nice as a mechanism, but what it means practically is that we could use this drug as a monotherapy. We didn't need a second drug. We don't have to worry about viral resistance, and viral resistance is the bane of our existence when it comes to developing drugs. Next slide, please. And so we had the problem mostly licked But there was one other critical factor that occurred, and that is EIDD 1931 had fairly low oral availability. And you may recall back in April of 2020, Gilead Sciences got emergency use authorization for a drug called remdesivir. And remdesivir is actually pretty potent against the SARS-CoV-2 virus, but it has to be administered intravenously. Now, think about what that means strategically. If you can only give the drug in a hospital setting, and our hospitals are overwhelmed with SARS-CoV-2 or COVID-19 patients, then it's very limiting in terms of the number of people that we'll be able to help with this drug. Also, the disease has two general components. One we consider the viral replication component, where the virus is just reproducing, and the other is an inflammatory response by our immune system. If patients get too far along, the viral replication part of that disease becomes less important, and the inflammatory stage becomes more important. So if you don't treat people early with antivirals, they're useless because what you really need is an anti-inflammatory. So the way we solve that problem is to develop an orally active prodrug of EIDD 1931. And the prodrug is simply something that is metabolized away to generate 1931, which then goes on to do its thing. And we were able to do that. And in order to do that, you have to have something that will permeate through the intestines. And that's a kind of necessary condition for oral activity. And by making a simple modification, we were able to do that. And now, instead of having to give these intravenously, you can give it orally. And that was what was missing all along in our armamentarium. So if you think about vaccines are, and they are amazing. They've done a great job, but they're all protective. They're all prophylactic, right? They all prevent you from getting the disease or minimize the consequences when you do get the disease. But they're not useful once you have the disease, because it takes the innate and the adaptive immune system too long to generate antibodies. That'll take maybe 8-10 days. They always say it takes 2 weeks to be fully protected with your vaccine. So you can't use it for someone who's infected. And this is where an oral antiviral comes in handy. And now molnupiravir, as it's called, is available. And my last slide just shows what the news release on December 23rd of last year. And this became the first first oral drug approved for the treatment of SARS-CoV-2 patients. So that's the story I like to tell you. And David, perhaps we can have some questions.
Thank you very much, Dennis. That was fascinating, and it's the journey sounds tremendous. We're going to ask the audience to start popping questions into the box so we can hear from them. I do have one or two questions for you. One has to do with sort of the business school side of this. Drive sounds like an amazing process, or the concept of Drive is tremendous, because it seems like you've been able to break down the monolithic nature of academia towards getting things done. Has that been? Is that something you're hoping to export to other schools, other universities around the country? Are other universities doing similar things, or is this really unique to Emory and Drive and you?
Well, it certainly was unique, and we've gotten a lot of attention, many questions from all over. And it's just a question of, in order to get it going, you need to fund it to get it going. And most universities, they may have a lot of money, but they don't have a lot of fungible funds. So it's problematic. So now, actually, with molnupiravir, we'll be getting quite a bit more revenue into Emory. And what we're going to do is expand DRIVE and our footprint to include oncology and neurological disorders. So we hope we'll be able to make an impact on a wider range of disease states.
That's great. I have a more personal question from one audience member that we always love to hear these types of stories. How did you decide for yourself and your career to specialize in drugs? What was the driving factor for you to go down this specific path?
So that's a funny question. I wasn't trained in this. I was trained in what we call synthetic organic chemistry, how to make things, but I didn't know any virology. And in fact, the last biology class I took was in 10th grade. In the late 1980s, the AIDS pandemic was killing so many creative people, and it looked like the pharmaceutical companies just weren't making any headway. They weren't doing anything, and I just felt like I had to do something. And when I told some of my colleagues about it, they told me things like, well, we used to think you were reasonably smart, but you're pretty stupid. How could your little academic lab compete with a major pharmaceutical company that's got hundreds of competent scientists and a lot of money, a lot more than you'll ever have? And the answer was, I selected a niche where I knew how to make those compounds that turned out to be emtricitabine and lamivudine. We had a very good way of doing it. And because we could actually run circles around all those big companies. So the key here is always, first, you have to be driven by passion. And my passion was, we had to do something to prevent the death of all these wonderful creative people. And secondly, you have to know what you're doing and select a viable path forward. And we were fortunate enough to be able to do that.
We have another question that takes us back towards your current antiviral, the work you're doing with antivirals. How will the basic building blocks of the antivirals that you've discovered be useful with future pandemics? Will we continue on that trend where, again, these antivirals will be able to continue to reinvent for a future pandemic? Because it looks like the concept of the pandemic is going to become part of our future forevermore.
Yeah, unfortunately, that's true. As more and more people live in cities, we're closer and closer together. So it's much easier when one of these infections starts out for it to spread. Remember the early days of Ebola, they might have been in West Africa and it stayed localized, but now we're likely to see more. So molnupiravir is the first drug for the next pandemic. That is, assuming it's a single-stranded RNA virus, we will have a storehouse filled with molnupiravir. And if it happens to be an RNA virus, we may actually be able to address it much more rapidly. But there are a number of other diseases that we don't have drugs for and that are being implicated. So Epstein-Barr virus, we don't have any drugs for. And there's some implication of this in MS and also in long COVID, that it gets reactivated. So we want to actually go after drugs or go after diseases where we don't really have any viable therapies, and these might have broader consequences than otherwise. So We will do this, and I think at least for a while, the government will continue to fund these until it people forget, and then but we're going to keep going one way or the other.
You know, your industry, or I'll say that I won't say it's your industry, the pharmaceutical industry. I'm not going to give you. The pharmaceutical industry has been under attack by many different fronts for many different reasons. Probably the most common one we've seen during COVID has been this accusation by the anti-vax movement about the safety regulations for drugs. So the question is, are safety regulations sufficient for mass consumption of what is being created these days? And how do we continue to educate the population to believe that they are safe for taking, since there seems to be this vax hesitancy movement that has really cropped up in a huge way over the last couple of years?
Well, first of all, we have to be able to believe in facts. And there are some that seem to push aside facts as they don't like those facts, or they're not consistent with whatever theory they're pitching. In the case of molnupiravir, before you could ever test efficacy, you have to test safety. And then, and finally, when we got to the phase 3 clinical trials, what we do in a trial is we list the so-called SAEs, the serious adverse events. Well, in the molnupiravir trial, the placebo arm had a higher number of SAEs than in the drug arm. And that says to me, all I need to know about the safety of this drug is that it's pretty safe. And that, we grew up in HIV where people had to take a drug every day of their life. And so if it was effective but poorly tolerated, people stopped taking it. And so I think this just brings safety to the fore and reminds people that it's not enough to be effective. It has to be safe and effective.
Great. At this point, I'd like to bring President Catherine back on the screen too. She has a question or two, and then at that point, she can help wrap us up. President Catherine, thanks for coming back on.
Absolutely. No, this is such a fascinating discussion. And I did have a question or two. Dr. Liotta, going back to some of the comments you made earlier, I'm curious back to your personal perspective on all this and your leadership style, because your lab has been such an innovator. What would you say are some of the characteristics you've used as a leader to foster that culture of innovation within your own lab?
Well, first and foremost, seek forgiveness rather than ask permission. Sometimes you just have to believe in yourself, and you have to believe in what is right to do, and you do it. The second is that no one individual is ever going to be capable of doing the kinds of things that we do. So it's very important with all of your coworkers to create an environment that allows each and every one of them to exert their own leadership potential. And so with smart people, I learned a long time ago, if you go in the morning and say, today I want you to do this, this, and this, that's not nearly as effective as saying, here's a problem. Let's decide on what our priorities are, and then you go off and see what you can do. And people like those kinds of challenges. They react positively to it. So let every member of your organization feel that they're in a position to make important contributions.
Terrific. Thank you. David, I see there's another audience question. Maybe we can, because it's similar sort of perspective.
It is. And I think you leaned into it a little bit already, Dennis. What is the most satisfying part of your work?
Well, I have a lot of satisfying parts. I love to teach. It's fun. I love to see the little spark in people's eyes when they finally get what you're talking about. I love training people and bringing them along. And as they go off on their own career, seeing them be successful. I have to say, in this regard, happens more than I ever thought it would. Going into a restaurant, sitting down, having the server say, "Excuse me, are you Dr. Dennis Liotta? I've had HIV for 20 years, and I've been taking your drugs, and not only am I alive, but I'm happy and thriving, and I always cry. I I can't help myself because the science is one thing and it's great, but when that personal interaction is just amazing.
Well, your career is something for the movies.
Yeah.
And I don't know if anyone's going to consider turning your story into something bigger, because it is an incredible story. We have one. Catherine, do we have time for one more question here?
Why don't you, let's one more audience and then I'll ask my last question.
Okay, great. When was Merck engaged as the partner to commercialize this drug, and how did you go about selecting a partner, or did they select Molnupiravir for you? I mean, how did the Merck relationship happen?
So the way it worked was there was an intermediary in between. Ridgeback was a biopharmaceutical company run by some hedge market people who understood biotech, and we went every year. There's a conference in San Francisco, the J.P. Morgan Healthcare Conference,
Right.
which is the biggest networking conference in biotech, and you go there, and I don't know, 1,600 people register, but 30,000 people show up for the meeting. And it's a big networking event. And you go around and you pitch it. And so my colleague, George Painter, who was the project lead in this, he pitched it to one of the leaders at Ridgeback. And they were impressed. They liked it. And they first licensed it. And after they licensed it, they got in conversations with a number of different pharma organizations, and they negotiated with Merck.
That's great. That JP Morgan conference is
amazing. Yeah.
Unbelievable.
Yeah.
Catherine, go ahead and close this out.
Last question, random question. Why do drugs always have such strange names? And I think about, it's probably something about the chemicals that they're made of, but then I think about other, quote, consumer products like food, which is also made up of multiple ingredients, but you end up with cornflakes or whatever. How do they get named? What's behind all that?
So every drug has two names. It's got a brand name that you see on TV, and it's got a generic name. And the reason, of course, we need a generic name is when you publish scientific papers, you have to refer, if you do it by brand name, the brand can change from country to country and all sorts of other things. But the names actually have some rationale. So notice Molnupiravir ends in vir. So it's an antiviral drug. And so there are certain suffixes and prefaces that are attributed to certain kinds of drugs. And this, I'll spare you the details, but this was actually named after the mythical hammer of Thor. And we were hoping we would strike a blow against the virus this way.
That is a perfect way to end, and a perfect example of the behind the scenes view that most people would never know about. But thank you so much, Dr. Liotta. David, as always, just fascinating. And I love and particularly inspired by the passion that you've shared with us behind your work and just the purposefulness of it. And it very much resonates with our Rotary Club culture. So thank you so much for joining us, and just a great discussion to both of you. All right. So just a quick reminder, next week we are off for President's Day. And then we're back with Dr. Walensky right after that, February 28th. And also don't forget to please